I'm going to talk about one of our projects in particular for this talk, which is looking at situational incontinence. And so I'm going to tell you a little bit about what that is and how we kind of started to try and investigate it. And then where it sort of comes into my sphere of interest is the involvement of the brain in situational incontinence, and then us trying to find a therapy for it. So what is it? So this is latchkey incontinence, key in the door syndrome. Everybody's heard about it. Most people have experienced something when they've, come home and suddenly they're like, oh no, I've really got to run to the bathroom. We get lots of people in our older women with incontinence saying that, they think they can hold it, then they see the toilet and they can't. They can have gone 10 minutes ago, but they still leak when they wash the dishes. They don't have any problems with leakage when they're out during the day. They come home, they see the front door and psh. And so this is obviously sort of something that is it's up here in the brain, right? There's certain scenarios that trigger the bladder to have urgency and leakage. And we think it's a conditioned response, right? So what we're positing that generally, like a lot of people don't want to go to the bathroom in public if they can make it home. So people spend a lifetime sort of like, oh, I'll make it home and get into the front door, and they really need to go. And then obviously with aging, as pelvic floor starts to get a little bit less toned, as our brain control gets a little bit less, everything's a little bit less good at what it used to do. That's when those situations really start to overtake the bladder and you get this urgency and leakage in the face of these triggers. So they're usually part of overactive bladder symptoms. People don't tend to like have that on their own. It's usually part of the symptoms that people experience. And studies found that like up to about half of people that have UUI experience that urgency in these situational scenarios. And it's often bathrooms. People see a bathroom, they see a sign for a bathroom, and suddenly they've got to go, they've got to run. Any sort of running water, they turn the shower on, they put their hands in to do the dishes. But there's a lot of places that are specific to a single person. We found in our studies that church is a big divider. We had a lot of people that had urgency in church and they got to sit down and they're like, oh, I've got to run. And other people that felt that that was a safe space they've never been to. So some of these situations are very specific to individuals. So how have we studied this? How do we recreate that? Obviously, like in neurodynamic studies, it's really difficult to sort of recreate these sorts of scenarios. So we decided to just show people pictures. And I actually collaborated with an addiction researcher that did a similar thing with addiction cues. So people who are smokers had craving in certain places in the bar when they picked up a beer, when they got in their car. And she would show pictures of those spaces and elicit cravings in the lab. So we decided to do the same with people with urgency incontinence. So we got people to decide which were their kind of safe spaces where they felt they didn't worry too much about their bladder, and then their urgency triggers, which were very often the front or the garage doors, washing dishes, etc. So we got people to go out and take photos of these places. And so our initial pilot study, we just had 12 women who had urgency incontinence, who said that they had this situational incontinence. And we got them to sit down and tell us all of those spaces, talk through which places did trigger that. And we did urodynamics and projected those photos on the wall and got them to rate their urgency. And they measured detrusor overactivity and leakage. And we found that it worked, basically. People, they had an increased desire to avoid, they had an increased worry about leakage, they had detrusor overactivity. So it wasn't just a sort of a worry, it was an actual physiological response. And a couple of people actually leaked when we showed them photos of their front door. And in contrast, some of our sort of safe photos did actually reduce the desire to avoid and reduce worry about leakage. So we figured we were onto a good thing. So we decided to try and work out, how does that translate to what's happening in the brain? How does seeing these scenarios translate to making the bladder have urgency and leak. So we did a follow-up study, and we to look at the brain areas involved in these situational triggers. So we had 23 women that came in that had situational incontinence, and we showed them their photos of their urgency situations in a brain scanner, in an MRI scanner, and we identified the brain areas that were associated with these urgency trigger and safe situations. And we found that with a full bladder especially, people really reported a big change in bladder sensation and reaction to these trigger spaces. But even on an empty bladder, they did, even within the MRI scanner, which is obviously a very strange situation. And we found that there were a number of brain areas that were more active when people were looking at these urgency triggers than their safe spaces. And they all seem to have a sort of a visual processing, a memory component, sort of like they were walking through that memory of being in that space. So we've got visual spatial processing, information processing, emotional stimuli, sort of that recalling of being in that situation. And also the motor area, sort of urination control, motor control of the pelvic floor, as soon as they are exposed to these images compared to their safe spaces. So the brain actually sort of is doing different things. It is reacting to these images. So we wanted to try and stop it. Like, how do we stop people from having these reactions to these situations? And it's quite difficult to sort of decondition these associations. And so we wanted to try a sort of multi-pronged approach. And so we did a sort of cue exposure technique where they got the photos of the triggering places, and we sat them in a calm room for about 20 minutes while we put these projected onto the wall in front of them, and we gave them a mindfulness technique. So we taught them to do body scan, which is a mindfulness technique that takes about 15 to 20 minutes. And it's basically somebody guiding you to think about your toes, acknowledge any sensation, and then sort of like without judgment, and move on to your knees, upper body, goes through your entire body, and you're encouraged to sort of feel what you're feeling, but without any judgment of that. And usually they don't include bladder specifically. And we did include bladder. We sort of said, acknowledge your bladder, move on, try not to focus, just included it. And we also did some transcranial direct current stimulation. So what we did was this is a technique using electrical stimulation of the brain that's really useful if you pair it with a task. So mindfulness, cue exposure, and it improves the effects of that task, and it sort of upregulates or downregulates, depending on the configuration, the region of the brain that you are pointing it at. It's very superficial. It's generally only cortical areas that you can get. So we chose to do the prefrontal cortex because that's involved in executive control avoiding, but also in the processes involved in mindfulness. So we did that study. We got women over 40 who had this situational incontinence. So we randomized them to have mindfulness alone, where they practiced it for a few days before and then practice it in 4 sessions in our lab, where they're looking at these cue trigger pictures on the wall. And so they came in for actually 4 sessions over a week. And one group had the mindfulness, the other group had TDCS alone without the mindfulness, and one group had both together, so mindfulness and TDCS. All groups were looking at their triggering situations on the wall while they were doing these exercises. And this is an image of the mindfulness. This was not the setup that we used. That was not where the anode and cathode were in our specific setup. We were stimulating the right dorsolateral prefrontal cortex so that positive red anode was on the right temple and the negative cathode was actually on the left bicep. So that was sort of trying to upregulate the activity in the dorsolateral prefrontal cortex. So did it work? Yeah, it kind of did. It did pretty well. More than 60% of participants reduced leakage by at least 50% in all interventions. 20% became dry after the intervention, and there were significant reductions in ICIQ floods at 4-week follow-up, which suggests that the that improvement continued for at least 4 weeks after they'd stopped, and they liked it. They scored it out of 100 as to how acceptable it was, and they said it was, they got 83 out of 100 for TDCS and 90 out of 100 for mindfulness. And we got some really good feedback. So a lot of people incorporated the mindfulness into their daily practice. We had people had to come in 4 times for an hour each day, so 4 times in a week. And out of all the people that started the intervention, there's 60 people, nobody stopped, nobody quit, everybody got all the way through the intervention, which is, I know it's research and we do have good participants, but that's still quite good for quite a high burden intervention. And so you can see here this is a visual of the changes in bladder diary. So we've got significant reductions in leakage episodes per day, and also triggered episodes. So they recorded a 3-day bladder diary, and they said which urgency, leaks or trips to the bathroom were triggered by a scenario like the front door, or one of their scenarios that they had actually identified. So we did get some pretty impressive reductions that were not dissimilar to that you get with drug or biofeedback. And we also did a measure of Curie activity. So we asked people to rate their urgency on viewing these images before and after the intervention. And you can see in the top ones, we sort of closed the gap between urgency triggers and safe spaces. So by the end of these interventions, they were not feeling any different looking at those images that were triggering than those that were safe. So that's another sort of evidence that it was doing something. So we found that all 3 interventions reduced the number of triggered episodes and leaks. And the combination of mindfulness and TDCS also reduced the number of urgency episodes per day. And they were really well liked and well received. So we feel like it's got good potential to be a non-pharmaceutical adjunct to therapy for urgency incontinence. Mostly because, especially in older adults, we don't necessarily like to give more pharmaceuticals or more invasive things. And this is a fairly low burden non-pharmaceutical intervention. It's non-invasive. It's a good alternative to a drug, and or you can do it with, right? It's a valuable adjunct as well. And it does have the potential for more generalized effects. If we're doing a mindfulness component, mindfulness has general effects on well-being. So what we're trying to do is find ways to improve continence through promoting brain mechanisms as one of the ways of supporting the continence mechanism in older adults. Obviously, we're not curing anything. We're trying to have multiple ways of tackling different aspects of urgency incontinence. So what's next? In this particular domain, we only looked at one brain area. What happens if we stimulate one brain area? So we're currently submitting a grant to look at multiple different brain areas to see if any of them work better, pair better with mindfulness. There's multiple different ways that you can actually give TDCS with different montages and different stimulation methods. And we kind of want to understand like how that works in the brain. And if we understand a little bit more of how that works, maybe we can sort of target this a little bit better. Yeah, and this is just thinking of like the body scan mindfulness as a task. There may be other behavioral interventions that can be paired well with TDCS. I know people have used TDCS with like pelvic floor muscle therapy and stuff. So it's quite a promising and fairly low burden way of doing this, and also has the potential to be a thing that you can do at home with sort of a bit more work on the intervention. This is our research team. So I've got for you.
All right. So my area of interest, my research area of interest is Nocturia. And as I was starting my fellowship and I was, planning to do my T32 and postdoc fellowship in clinical research, I was interested in lower urinary tract symptoms. And as I started seeing more and more patients in this cohort, in this older age group, one recurring theme was always nighttime awakenings to void. Sometimes patients would bring it up themselves, saying that it's a problem, but while other times it would be more on review of system. And I asked, and how's the sleep? And it turns out that sleep is very disrupted, interrupted, with frequent awakenings to void. So first few slides, I'm going to breeze through the definition of nocturia by ICS. As we all know, it's need to awaken more than one time a night. It has to be preceded and followed by sleep. Nocturia is one of the most common of all the lower urinary tract symptoms and one of the most bothersome. And a customary epidemiology slide, we know that nocturia increases the prevalence increases as we age and in both genders, regardless of bladder outlet obstruction or overactivity, the nocturia prevalence increases in both genders with as high as over 33%, over 35% of both genders will have at least two awakenings a night to void, which is a more reliable bother symptom of nocturia rather than just one awakening per night. But as I was learning and reading more about nocturia literature, I would dive into literature. It would be more of urology presented data, but there is this big overlap for sleep. And I was wondering, where is sleep in all these? Where is sleep research with all these bladder symptoms? So how are they addressing this? And I found this older study in sleep, which looked at 8,900 participants in all age group 18 and over through a telephone interview, trying to find out, the characteristics of sleep disruption in different age groups. And of course, they found that, as these people age, the nighttime awakenings become more and more frequent, and the highest propensity was seen in age group over 65. Mostly what they noted was that people who were waking up almost at least more than 3 to 4 nights a week, they were having these issues as a more chronic, and 90% of these people waking up more than 3 to 4 nights a week would say that these symptoms have lasted for over 6 months. And when they probed further to look for the reasons of these nocturnal awakenings by group, and this is just a very small list of all the causes of these nighttime awakenings that these telephone interviewers asked for. Of course, the top of the list was going to the bathroom in all age group, but as you can see in older adults, upwards of 75% of the people reported these nocturnal awakenings to go to the bathroom as the most common cause of these nocturnal awakenings. But when you talk about sleep, and I was working with some sleep researchers at that time, their concept was that, people have nocturia, they have bladder issue, the bladder is waking them up, and that's causing these sleep disruptions. So when we talk about these etiologies of nocturia, one thing that is a recurring theme in all these papers and discussions is that it's multifactorial. There are multitudes of systems in the body that contribute to nocturia. They are worsened by nocturia. There is bidirectional relationship. It's multidirectional. There are so many other comorbidities that contribute to nocturia. So every time we talk about nocturia, we talk about the multi-complexity of the disease process. So trying to simplify it in my head so that I can focus on the disease in a more nuanced fashion and take my research forward, I try to put nocturia etiology in 3 main buckets. One is increased production, then there is difficulty with storage or some changes in storage, and then one of the bigger buckets is sleep. And as I said, when we talk about sleep, researchers, they think that, it's the urine production and it's the bladder issue that's causing these sleep interruptions. And to some extent, they are right, because one of the most common causes of nocturia in older adults is nocturnal polyuria, where people are producing more than, sometimes in excess of 50% of their 24-hour urine volume during their sleep time, during their sleep period. And if they have all that urine production, it does wake them up at night to go to the bathroom. It does cause sleep interruption and promotes these nocturnal awakenings and insomnia in older adults. So that made us wonder, like, why are older adults producing more urine at nighttime? What we generally know about urine production is that, it does follow a circadian rhythm. And there was this study, in a circadian study, in which they recruited healthy young and healthy older participants. There were 26 young men and 23 healthy older men and women. It was a mixed study. And they brought their participants to a research lab, and the participants stayed there for 40 hours. The participants remained awake for those 40 hours under constant routine protocol. So they were in semi-recumbent position. They were given food and fluid at equal intervals, and the effect of light was taken away. And what they noted was that black is the urine production in younger folks, and red is the urine production in older participants. And what they showed is that in this circadian study, under constant routine protocol, older adults produce more urine during nighttime and less during daytime compared to their younger counterparts. So they said that, there is some changes in circadian rhythm of urine production even in healthy older adults, and that's why they're producing more urine at night. And that perhaps is one of the reasons why they have nocturnal polyuria. But is that the whole picture? Now, what we know about the regulation of urine production is that there are two antagonizing systems that are acting on kidneys to regulate urine production. One is this vasoconstrictor sodium retaining system, which is arginine vasopressin, or AVP, or antidiuretic hormone, or ADH. So one is ADH AVP versus, and the other is RAS system, which is renin-angiotensin aldosterone system. And then there is diuretic vasodilator natriuretic system, which is arterial natriuretic peptide. So it's the balance between these two symptoms that kind of manages water homeostasis, sodium homeostasis, and all the electrolyte management in all humans. So in this study, this was a very interesting study done, as you can see, way back in 1999, where they had healthy young adults, and they changed the sleep time. So if you look at the top column, top row, they allowed the participants to sleep during the sleep hours between 11 p.m. and 7 a.m. and measured the hormone levels in these younger folks, and then brought them back to the research lab and changed the sleep time, where they were awake overnight, and they allowed the participants to sleep between 7 a.m. and 3 p.m. and they measured these hormones. The last column right here is cortisol, where they say that, it's a circadian hormone, which is tightly regulated by endogenous rhythm, not affected by sleep. And they said that, yes, when you change sleep time from nighttime to daytime, cortisol still peaks when it peaks, which is the morning hours. So it is the endogenous rhythm of the hormone. However, when they looked at aldosterone and renin, they clearly saw that sleep definitely significantly affects these hormonal secretion, hormonal peaks. And as the sleep time shifts, the peak time shifts along with that sleep. So this was a very convincing evidence that, as sleep time changes, these hormonal peaks of aldosterone and renin changes with that. But the question is that these were the studies done and there are more than one studies that have shown this. This is just one example that I am displaying here. But the question is that, this is done in younger folks. How about older adults? Do older adults reflect the same pattern of hormonal regulation as they age? So we did a study where we recruited healthy older adults with no sleep issues, with no bladder-related issues, had no nocturia at baseline, and we brought them to our research center for 2 24-hour protocols. For the first protocol, the participants were allowed to, they would come at 7 a.m. to our research lab. They were allowed to, they stayed in our research lab for full 24 hours. They were allowed to have daytime activity as they would, and they would go to bed at their habitual sleep hour. 6 to 8 weeks later, we brought back the same participants, and it was in constant routine protocol, what I had just explained in the previous study, where they stayed awake. It was constant wakefulness, semi-recumbent position, and the diet was replaced by supplements at regular intervals. We drew blood specimen from these participants every 4 hours during the daytime, and then every 30 minutes from 7 p.m. to 7 a.m. And we looked at these renal regulatory hormones to see how these hormones levels change as the sleep is taken away and they are in constant routine. So the first graph here is melatonin, which was our positive control. Of course, melatonin is, again, a circadian hormone with endogenous control, and melatonin secretion did not change in either of the two protocols. The green line indicates habitual sleep-wake protocol, and the red one indicates constant routine protocol. However, the other hormones that we checked, AVP showed a significant change where the peak happened during sleep time, but when for the same individual, when sleep was removed, there was no peak in these hormones. Similar changes were seen in aldosterone, where the nighttime peak dissipated or was attenuated in constant routine protocol. In ANP, we saw that there was a steady increase in ANP in normal sleep-wake protocol. However, that steady increase dissipated during the constant routine protocol. So what we concluded was that in healthy older adults during basal condition, ADH and aldosterone, they peak during sleep, followed by a steady rise in ANP towards the early morning hours. And what happens is, as these antidiuretic hormone and aldosterone are peaking, they contribute to sodium and water reabsorption, leading to decrease in nighttime urine production. And then as early morning hours approach, ANP rises, which has an antagonistic effect on the RAS and leads to natriuresis and diuresis. And then, patients wake up and they go to the bathroom and they void. But all these diurnal rhythms were lost with loss of sleep and posture control. So does it mean that, in older adults with chronic insomnia, does that has a role in changing the milieu of these hormonal secretions overnight versus daytime? And does that contribute to nocturnal polyuria? And should sleep be a target not only to address these frequent awakenings attributed to perhaps insomnia, but also to address nocturnal polyuria to change the way urine is produced, the nighttime regulation of urine production in these older adults? And is it just as simple as adding desmopressin, which is not true, because desmopressin works only on the water metabolism, and absorbing water without this, sodium balance with that does lead to increased side effects, especially in frail older population. So coming back to our same picture of this sleep and production bidirectional relationship, now let's move to the other aspect of it. Is insomnia itself affecting these nighttime awakenings, which is something that sleep literature has looked into? We all know that as we age, our sleep changes. Sleep in older adults has declined after the age of 60 to 65. There is a clear decline in deep sleep stages. There is clear increase in frequent awakenings through the night, and causes these sleep interruptions. Now, with these sleep interruptions, older adults are vulnerable to have insomnia, and any triggers like any stressors, any illnesses, they can act as a precipitating factor where with vulnerable sleep changes in older adults, they push them, these illnesses can push them into insomnia. And there have been electrophysiological and neuroimaging studies that have demonstrated that patients with insomnia have nighttime hyperarousal state. And in this hyperarousal state, they are more sensitive to bladder fullness. And as they're more sensitive, they wake up more frequently at night to go to the bathroom. And this nocturia then perpetuates the cycle of insomnia and hyperarousal, which becomes a vicious circle in patients, in our patient population. So is the sleep only affecting the nighttime, voiding nighttime bladder sensitivity and nighttime urine production, or is it affecting storage in any which way? Now, this is an area of research that hasn't been much looked into, but it just entices me, it interests me a lot. And so I'm just throwing it out for the group to consider and have their thoughts, share their thoughts, in this meeting. So in general, bladder is considered, it's perceived as a reservoir of urine with tight junctions, and that does not affect any water metabolism. And the main water metabolism is considered to be through AVP, which is the main hormone. And we all know that it targets V2 receptors, which we know are there in renal collecting duct. And the way ADH works on these renal collecting duct is through this aquaporin, which is a member protein that mediates like rapid and selective water transport across cellular bilayer. So it's always considered that, these aquaporin channels through V2 receptors mediated by AVP are the main mechanism of water metabolism. And there are diverse tissues in human body that have demonstrated these aquaporin channels, of course, kidney, but also brain, etc. However, there is more in situ and in vitro studies that have shown that aquaporins are expressed in bladder human urothelium as well, which makes us wonder what's the role of these aquaporin membrane protein in human urothelium. There is some data to suggest that they could be contributing to some of the water and AVP changes in overnight urine excretion. This was a study done in little children, in kids aged 9 to 11 with nocturnal enuresis. And these were like primary monosymptomatic nocturnal enuresis. The kids had no other bladder issues other than they had nocturnal enuresis, and they were compared to 22 healthy controls. And what they found was that AVP in normal children had this characteristic circadian rhythm where it peaked during the evening hours in the nighttime. But children with nocturnal enuresis, they express significantly lower levels of nighttime serum AVP, which correlated with urine aquaporin 2, as well as urine osmolality. So the authors concluded that these enuretic children tend to release lower amounts of aquaporin 2 as a result of low AVP stimulation. And, not to draw parallels, but we do see that sleep deprivation does lead to low AVP in older adults, as we had seen in our study previously. In another study, this was a very interesting study that was in which they included 24 healthy volunteers aged 11 to 50, but as you can see, 70% of the participants were aged between 20 and 39. What they did was they did overnight bladder capacity assessment in these participants using transabdominal real-time 3D ultrasound. This was a study that was conducted at participants' home. They were asked to empty their bladder before going to bed, and they would take 300 to 500 ml of water at least 30 minutes before going to bed. And in these 24 healthy volunteers, the study reported 41 events where they recorded overnight nighttime bladder volume in these participants with this protocol. And what they found was that, of course, functional bladder capacity was the most important factor that controlled the capacity of urine in bladder while these participants were sleeping. And the urine production that they saw fell in two main categories. One they call as gradual elevation pattern. And as you can see in the left panel of these graphs, which is gradual elevation pattern, where overnight urine production from kidneys, it just gradually increased through the night, did not reach functional bladder capacity till early in the morning. And the subjects were awakened in the morning with sensation to void when their bladder was full, and they would go to the bathroom and empty their bladder. This was noted in 54% of their participants, but 46% of their participants had what they called steep flat pattern, where within an hour of going to bed, the participants had a steep rise in urine output. And then it kind of plateaued throughout the night. Now, they did not differentiate the characteristics of these participants. They were all healthy participants. So we don't have much data to say what was the difference between these two participants, but clearly there were two different patterns of urine production. Interestingly, in steep flat pattern, when they were producing excess volume in the first half of the night or early part of the night, and then it plateaued through the night, it makes you wonder, urine is being produced by kidney. Kidneys never stop producing urine. So where is this volume going? How is bladder maintaining the constant volume through the night? Interestingly, in these participants that had steep flat pattern, they reported what we call TVR, what they call TVR, which is basically temporary volume reduction, where bladder would have some volume and then there would be a reduction and then there would increase again and then there would be reduction again. Now, these volume reductions were valued between, like the volume varied between 50 to 120 ml. This was more common in participants who had this steep flat pattern, but 9% of the episodes happened even in gradual elevation pattern, which makes us wonder that, there is some water metabolism that's happening overnight as the person is sleeping and the volumes are changing throughout the night. This was another interesting study. It was presented as an ICS abstract. Full paper was never published, but in this abstract, they included 8 healthy undergrad students, and the students completed a bladder diary before the start of the study. And then the study protocol included that day 1 and day 2, the participants were asked to void every 3 hours between 8 a.m. and 11 p.m. Overnight volumes or overnight voids were not recorded. So it was, they would have every 3 hours, and then there was a rest day in between, and then day 3 and 4, they would void every 5 hours. The participants were asked to keep a record of intake and then, of course, a bladder diary output. Interestingly, the fluid intake did not significantly differ by report in these participants in the 3-hour protocol versus the 5-hour protocol. However, the volume voided was significantly different. So, which again brings the question, this is more of a pictorial representation of what they were saying, that, in 3-hour protocol versus 5-hour protocol, the volume decreased. And in addition to the volume, they also saw, they also reported a decrease in sodium, potassium, and urine osmolality in these participants who were holding the urine in their bladder for longer duration versus shorter duration. So all these, these are smaller studies. They don't have any conclusive evidence, but they do warrant further research. And they do raise question about role of bladder and urothelium as passive storage versus are these cells playing an active role in contributing to water homeostasis and electrolyte transport. What is the role of these aquaporin in this water transport? And does sleep and AVP, how sleep deprivation leads to decrease in AVP secretion overnight, does that have a role in affecting these bladder storages? So I'm going to stop here.